This is Module 9, Fatty Acid Metabolism. In this video 9-1, an overview of fat metabolism, I'll be talking about the different ways in which fatty acids are moved throughout the body for fatty acid metabolism, review the structure of a triacylglycerol, which is a storage fatty acid, and talk about the different enzymes that break them apart into free fatty acids. There are three sources of fat in a vertebrate diet, or in a vertebrate organism. You can obtain fats in the diet, and 40% or more of the daily energy requirements of people, or humans, are supplied by dietary fat. And so that I'm going to talk a little bit about how dietary fat is processed by our bodies. You can also have an endogenous source of fat where you're mobilizing fat from stored adipose tissue. I'll talk about that in video 9-2. And then if you're consuming excessive dietary carbohydrates, the liver will actually convert those carbohydrates into fatty acids as a way to sort of hang on to and rescue that high energy molecule. And so remember that the liver can store carbohydrates in the form of glycogen, but there is an upper limit to how much the liver can store glycogen. However, there's no upper limit for how much fat you can store. Your adipose tissue will expand to allow for storage of fats almost indefinitely, to a point, obviously, but pretty fairly high ability to store fat. And so your liver, recognizing glucose as an important dietary source of energy, if it has already stored up all the glycogen that it can, it will convert dietary carbohydrates into fatty acids and then export them to adipose tissue for storage. And so a lot of people ask me, is it true that sugar turns to fat? And in a way, yeah, it is. If you consume excess sugar, you will gain weight in the form of fatty acids because that sugar is converted into fat by your liver. So when you consume fat in your diet, fat is a nonpolar molecule. They're really insoluble in the aqueous environment of the bloodstream in an interstitial fluid, which bathes all of your body tissues. So fats are very large, clustered together nonpolar oils. And so the first thing you have to do in order to solubilize them and absorb them through the intestinal lining is to break up these large fat globules into smaller ones through the use of bile salts. Remember, the lining of the intestinal cell wall, or the intestinal cells, are polar. They're made up of glycerophospholipids. And so those very polar heads are going to repel non-polar fatty acids from crossing over into the cell of the intestinal lining. And so first we have to find a way to make them a little bit more soluble so they can get across that barrier. So bile salts are produced and released into the lumen of the intestine, and they will combine with fatty acid molecules to break up really large fat globules into smaller fat globules. Bile salts are cholesterol derivatives, and so this is what bile salt looks like, and you can see that it's got a similar structure, the four-ring structure, to cholesterol. And so cholesterol, which is produced in the liver, can also modify cholesterol, or the liver can also modify cholesterol to produce bile salts, and then those are exported and stored in the gallbladder. So after you eat a fatty meal, the gallbladder will release the bile salts that the liver made into the small intestine, and they will mix with fatty acids through the formation of micelles. And so a micelle, remember, is sort of like half a lipid bilayer, where you have one layer of glycerophospholipids with phosphate polar heads sticking out and non-polar tails in the core. And so fats will mix with those because they prefer to mingle with the core, which is non-polar tails of the micelle. And then the micelle's polar heads will surround the fatty acid molecule, which makes it more soluble in this aqueous environment. Oh, I'll come back to what a lipase is in a moment. Okay. So that's how we absorb exogenous fatty acids that are dietary fats. We can also store fatty acids in adipose tissue and mobilize those fatty acids when fuel stores are low and there's no glycogen left. So your body will use all of the glucose it can before dropping the blood glucose level too low. Then the liver will start to break down glycogen and dump glucose into the bloodstream to maintain glucose levels. But when the liver runs out of glycogen, that's when we start to mobilize fatty acids from our fatty acid store. And so mobilization from the adipose tissue is triggered by the release of glucagon, which, if you'll remember, is a hormone that is released when blood sugar starts to drop and you've gone quite a long time without eating. Epinephrine is another molecule or another hormone that triggers mobilization of fatty acids. It's your stress response molecule. And so during times of acute stress, so like the fight or flight response, epinephrine will release virtually all high energy molecules into the bloodstream so that all of your muscles can utilize it for energy to get away from your attacker or fight off your attacker. Anyway, so the release of fatty acids from fat stores involves an enzyme that responds to glucagon and epinephrine. So when glucagon and epinephrine are released, that will activate this enzyme called a hormone-sensitive lipase, which will start to cleave fatty acids from the adipose tissue and release fatty acids into the bloodstream. So when fatty acids are mobilized, they're transported to different tissues that's capable of using them. And so the muscle and the heart can use them, and oxidize fatty acids for energy production. The brain cannot use fatty acids, so that's why that's missing from here. The brain does not use fatty acids as a source of energy. Only glucose can be used by the brain. Only the brain can use glucose, sorry. There's another carrier molecule in serum which binds to free fatty acids. So these lipases will break off bits of fatty acids from adipose tissue and release free fatty acids into the bloodstream. But remember, fatty acids are long hydrocarbon chains, which are really nonpolar and don't really like hanging out in the bloodstream. In fact, if that happens, they start to cluster together and you run the risk of forming clots in the bloodstream. And so there's a protein called albumin where fatty acids can bind to it and it makes it more soluble and prevents them from clustering together. And so that helps to transport fatty acids, free fatty acids in the blood. There are other carrier molecules, which I'm going to talk about, but that's one of them. Let's come back to this idea of a lipase. So I talked about a hormone-sensitive lipase, which helps to break down cellular fat stores in your adipose tissue. And these are activated by glucagon or epinephrine. But there are two other types of lipases that help to absorb fatty acids from the diet, from exogenous sources. They are pancreatic lipase and lipoprotein lipase. Actually, this one can kind of do both. Okay, so the intestinal lumen, when you consume fat, some of those fats can be broken up within the intestinal lumen, ones that are absorbed through the intestinal wall. And so fatty acid TAG digestion, breaking up triacylglycerols, begins in the intestinal lumen. So it's been mixed with bile salts, it's soluble, and then it will mix with pancreatic lipase, which will break fatty acid chains off of a glycerol backbone that makes up the TAG. In the walls of the capillary, this is where you'll find lipoprotein lipase, which helps to absorb fatty acids from lipoproteins, which is a transport molecule found in the blood. And so these transport molecules can either pick up dietary fat or help to mobilize stored fat. And so that's why I said this one kind of does both. Dietary fatty acid digestion and endogenous fatty acid digestion. So more on lipoproteins in a moment. Pancreatic lipases specifically within the intestine will bind to a TAG, break off the one of the carboxylic tails. And so we'll do this sort of systematically. So one is broken. So this was one reaction where lipase bound to this one here, broke this ester bond, and then released one free fatty acids, leaving behind a TAG that still has two fatty acid chains attached to it. And so the lipase enzyme would have to repeat this reaction to free all three fatty acid chains. So this is happening in the intestinal lumen. TAGs are broken down, fatty acids are produced, mixed with bile salts, and so then they absorb through the intestinal lining. Now we're inside the body, on the other side of the tissue barrier, and so we're headed for the capillaries in the bloodstream. Again, fatty acids can combine a little bit with albumin to transport through the bloodstream, but there's a limited amount of serum albumin. There are other specific proteins that can transport fat between different organs specifically and bind to specific organs to give them the fatty acid energy that they need. These proteins are called lipoproteins. Again, I'll talk all about lipoproteins in video 9-2. So I talked about pancreatic lipases here. I'll talk about lipoprotein lipase in the second video. And again, hormone-sensitive lipases convert tags into fatty acid groups and also the glycerol backbone in the presence of glucagon and epinephrine. And so when a lipase breaks up a triacylglycerol, any lipase, it's going to generate a glycerol backbone, which can be converted into intermediates of the glycolysis pathway. And so we can get a little bit of energy. About 5% of the energy generated from oxidizing a fatty acid comes from the glycerol backbone and funneling into the glycolysis pathway. The remaining 95% of the energy that can be oxidized and used to produce ATP is coming from beta oxidation of the 3 fatty acid groups that are released from the backbone when lipases cleave them off, cleave these ester bonds. And so now we have these 3 fatty acid groups that will go through a special metabolic pathway referred to as beta oxidation. I'll talk more about that pathway in video 9-3. And so I've talked about there are exogenous sources of fats which are coming from your diet, and there are endogenous sources of fats which are coming from your stored adipose tissue. And so this is a summary of the 2 pathways for for either mobilizing stored fatty acid, absorbing dietary fatty acid, and then transporting those fatty acids via lipoproteins throughout the bloodstream. So I'm going to come back to this image when I talk about lipoproteins in video 9-2. But just to summarize, here we have fatty acids that are cleaved via pancreatic lipase off of tags. And so where are we starting here? So we're in the intestinal, so we've consumed dietary fat. Here we are, we start here. We consume dietary fat, enter the stomach, that breaks up some of that fat into large droplets. That empties into the small intestine, where those large lipid droplets mix with bile cells to form smaller micelles. Pancreatic lipase will then bind to the tags in those micelles, break up free fatty acids, and then free fatty acids, which are smaller, can absorb through the intestinal lining, through the cell membrane. Within the intestinal epithelial cells, they're rebuilt back into tags and mixed with special transport proteins called chylomicrons, which then enter the bloodstream. I'm going to talk about the chylomicrons in video 9-2. Once they enter the bloodstream, their destination is the liver. And then the liver will process them for either storage into the adipose tissue or transport to other tissues that can use fatty acids as a source of energy. And so this blue arrow represents the exogenous pathway, dietary fat into the blood, into the liver. These red arrows represent the endogenous pathway. And so the liver will take any of the tags delivered to it by the chylomicrons and repackage them into a lipoprotein called VLDL. That enters the bloodstream, and so if the destination of this molecule is to store it, we will store it in adipose tissue. So lipoprotein lipases will break them up and store them in adipose tissue, or capillary lipases will break them up, or lipoprotein lipases in the capillaries can break them up and allow for muscle cells to use those as a source of energy. And I'm going to talk more about these other lipoproteins in the next video.